Hi everyone, welcome to Madness and More. Today we're going to be going through osmosis and focusing on the cell membrane. So I am considering this chapter 5 because of the order that I'm going through in these lessons. It is going to be very similar to your Princeton Review or Barron's College Prep books. The order that I go through in. The chapter numbers will not be the same though. Um, Another key point to remember while learning from these video series is that if you ever have any questions, you can feel free to fill out the contact us form or email me at mathisandmore@gmail.com to get in touch and I can help you with any doubts. So let's begin with the lesson. Before we begin, let's review the laws of thermodynamics or energy transfer. The first law states that energy cannot be created or destroyed. It can only be changed from one form to another. The second law of thermodynamics states that energy transformations increase disorder or entropy, and some energy is lost as heat. Here you can see the energy being transferred. So this slide is not something that you're going to need to know. So what I mean by this is for the exam, you do not need to know the formula, just know the concept. So energy is the capacity to perform work. And all organisms require energy for life functions. So there's kinetic energy, which is motion energy, you can remember. So this is the energy that's being used when a bike rider is going downhill, for example, like you see here. And potential energy is stored energy. So something like what you would see here, the biker is on top of a hill. They have potential energy. They have stored energy that can become kinetic energy. So which reactions need an input of energy and which ones don't? So these would be the spontaneous reactions. So an easier way to kind of understand this is that the change in G, so G is the change in free energy. So that gives energy formula you do not need to know, but I'll go over the concept. So G is the free energy, the change in free energy you have. H is the total energy that is available. This is known as enthalpy. And then J is a change in the system's entropy. And then T is the temperature in Kelvin units. So a lot of bio formulas will have Kelvin. So an easier way to understand the concept is that the change in G is the difference in free energy from the reaction start to finish. An example of this would be delta G, which is the change in G, is equal to the final state of G minus the initial state. So in the images here on this side, you are able to see that if I can get this to, okay, so here we go. So here you can see the starting energy, it goes up and then it goes back down, and then this is your product. So this is your kind of ending energy, starting energy, ending energy right here. Same on this side, on the bottom, the starting energy, ending energy. So we will understand, we'll look into this in more depth in further slides. So if delta G is negative, that means the reaction is spontaneous and that means that there is a loss of free energy. And if G is positive or 0, that would mean that the reaction needs energy input to occur. So if the result of the equation, of this equation, is negative, then it is going to be a downhill reaction. So it would look like this. If I can just get this to work, yes. So the graph highlighted in blue is what it would look like if the equation is negative, if the result of the equation is negative actually. And then if the result of the graph is positive, your graph will look like the graph being highlighted in purple. It will go upward. Let's look at the energy in the cell connection. This is a continuation of the last slide. So free energy, which is G, is a measure of a system's instability. There is a tendency for systems to change from an unstable state to a stable one. And the change, if G is going upward, if G is increasing, then that would mean that it will at some point go down, you know. So the state of max stability is equilibrium, and G is the lowest when the reaction is at equilibrium. A reaction is spontaneous and it can do work only when moving towards equilibrium. So here's an example. The diver is standing at the top of the diving board right now. This diver, they are at a very high amount of instability. They are not stable at all. As they're going down, they are moving towards stability, because when they hit the pool, suppose the pool is right here, when they hit the pool, they're no longer stable. They're no longer unstable, which means that this is equilibrium. This is equilibrium for the diver. So when the diver will reach the pool, they will be out of energy, because when you reach equilibrium, you are out of energy. So the diver will not have energy when they hit the pool here. So let's look at the chemical reactions that happen in our bodies based on the change in G. So an exergonic reaction is when energy is going outward or energy is released. So essentially it's when energy leaves. And this is going to be a negative delta G. Because the change will be negative and the reaction will be spontaneous. The greater decrease of negative delta G, the more energy is released and the more work can be done. Respiration is an example. We will look at cellular respiration in more detail in chapter 6, but that is an example of an exergonic reaction. So in respiration, your change in G is equal to negative 686 kilocalories of glucose. And that would mean that 686 kilocalories are given off when glucose is burnt into CO2 and H2O. An endergonic reaction is energy being put in, and it's when something absorbs energy. So this would be a positive change in G, if I can get this to work. Yes, it would be a positive change in G, and the reaction would need energy input. energy storing reactions are not spontaneous, and the greater the delta G, the more energy is stored. So photosynthesis is an example of an endergonic reaction. We'll go into more details on photosynthesis in chapter 7. So delta G is positive 686 in photosynthesis. 686 kilocalories or mole, which is the molecular weight of some material in grams. And then the so that would mean if the delta G is equal to 686 kilocalories, that would mean that the product, which is glucose, has 686 kilocalories or moles of stored energy. So a good question is, where did the plant get the 686 kilocalories to build one molecule of glucose? So how that works is that the plant will perform both photosynthesis and cellular respiration, but the plant gets energy. It gets all of the energy from the sun. So the sun is the main source of energy for the plants to get all their energy from. Now let's look at metabolism, which is the sum of all your endergonic and exergonic reactions. Cells carry out thousands of chemical reactions a day. They usually build in the night and they break up molecules in the day for energy. So coupled reactions are how cells use endergonic reactions and exergonic and endergonic reactions together. So they use exergonic reactions to fuel the endergonic reactions, because remember, exergonic reactions put out energy while endergonic reactions take in energy. So how this is done is it's done by manipulating ATP, which is adenine triphosphate. So the A, the T, the P are taken from ATP, adenine triphosphate, and from adenosine triphosphate it becomes ATP. It's just a short form. And so by manipulating ATP, it powers nearly all forms of cellular work. The energy in ATP is in the bonds between its phosphate groups. And the energy in ATP is in the bonds between its phosphate groups. That's very important to remember because it's dependent on how energy is taken from ATP. So when you hydrolyze the ATP, it becomes ADP. So adenosine diphosphate is equal to ADP. So you might read ADP in a lot of books. So what happens is that the phosphate molecule is broken up. So you see that there are 3 here, and it's broken up into 2 and 1 and ADP. And this is used for energy. So ATP needs to break apart to essentially get energy. So now let's look at how ATP coupling works. ATP hydrolysis is an exergonic reaction that drives an endergonic reaction through phosphorylation. Phosphorylation is when things change shape. So a phosphate is pulled from ATP and is transferred to another molecule, making it unstable or reactive. And then this is usually done through a change in shape And so now the original endergonic reaction can take place because the overall change in G of the two reactions is exergonic. Chemical, mechanical, and transport work all accomplished through ATP hydrolysis and molecular phosphorylation. And cellular work can be sustained because ATP is a renewable resource that cells can regenerate. So we will learn this in more depth, but ATP is generated through cellular respiration. One muscle cell uses and generates about 10 million ATPs per second, which I think is just crazy. Let's look at how enzymes function. Enzymes are catalysts, and they reduce the input of energy, so that the reaction can take place faster and be cheaper. So this would be the energy, the activation energy is decreased. So we see that without the enzyme, I'll do that in blue. Without the enzyme, you have to put in a ton of activation energy all the way up to here. It's just to activate your reaction. But with the enzyme, I will do this in orange. You don't need to go up that high. You don't need that much activation energy. In total, there is a huge difference between the activation energies of them both. So each enzyme has a specific substrate. Um, so a specific enzyme, which is a protein, catalyzes each cellular reaction, and they have a unique three-dimensional shape, so that they can fit into their specific substrate. Their shapes then determine their functions and which chemical reaction they can perform. And they are all related to their 3D folding pattern, which is coded in the DNA strands, which then form your amino acids. And amino acids, as we know, when they get together, they form a polypeptide chain, which becomes a protein. Factors which affect enzyme activity is what we're looking at next. The temperature, concentration, pH, and inhibitors play a major role. This is a very commonly occurring topic on the FRQs that I practiced. So that's what I noticed. But enzymes are an important part of the AP biology course and understanding enzymes. So this is very important. For optimal function, some enzymes require non-protein cofactors. This includes a lot of vitamins. So that's why vitamins are very important. Metal ions or organic molecules, called coenzymes, often work as cofactors. Enzyme inhibitors can also alter enzyme function. For example, a competitive inhibitor blocks the active site, and so the substrate can't attach and remains unchanged. So this is used by medical companies and things like antibiotics to block things, to block bacteria that aren't in your body. So just to see how the normal enzyme works versus a competitive inhibitor, we see that in the normal enzyme you have a substrate, and it is the perfect shape to fit into our active site right here. But in the competitive inhibitor, this is the substrate. It would have fit into our active site, except we have the competitive inhibitor here, which goes in and takes the place of the substrate. And then we have the non-competitive inhibitors, which alter the enzyme's function by changing its shape. So we can see that the active site is still empty. Even if the shape is changed for the substrate, it is empty. But the non-competitive inhibitor that we see here has changed the shape of the enzyme. Many poisons, pesticides, and drugs are enzyme inhibitors. Now let's look at the structure and function of the cell membrane or plasma membrane. The plasma membrane is selectively permeable and controls the flow of substances in or out of the cell. Selectively permeable means that some particles can get through but others cannot. For example, small nonpolar particles can get through easily, whereas bigger polar molecules have a struggle to. So membrane phospholipids form a bilayer, as you can see here. This is a bilayer, and the phospholipids are made of hydrophilic heads and two hydrophobic tails. So I'm going to highlight one phospholipid. So this right here is just one phospholipid, right? And then I will shade in the head. So this is the head up here, right? That's the head. And then our tail is right here. I'm gonna get red. Right here is the tail. So that's just one phospholipid. And there's two tails. So you could see a tail 1 and a tail 2. And a more focused image is given here of how it's built. So phospholipids are the main structural components of the membranes, and the bilayer has heads on both surfaces and tails in the middle. This is because the heads are hydrophilic, so these like water on both sides of the phosphate bilayer, while the hydrophobic tails are not the biggest fans of water. They are hydrophobic, which you can remember as scared of water. What you're seeing here is a fluid mosaic, which is the model of the plasma membrane. So proteins and other molecules are embedded in the phospholipid bilayer. We have glycoproteins, which help with cell-to-cell recognition and are like the ID tags. You can see a glycoprotein right over here. I'm going to highlight it in red right here. And then we have glycolipids. Glycolipids help with membrane structure and stability. You can see one of the glycolipids here. It's like a little chain. And then we have oligosaccharides, which are the short carbon chains of glycocalyx. Glycocalyx are glycolipids and glycoproteins of the cell surface, and cholesterol is only found in animal cell membranes, and it's for stability. So proteins are like the functional units of the membranes. Proteins function in transport. They work as Gates, channels, and pumps. They help with enzymatic activity. They help with signal transduction, intercellular joining, cell-to-cell recognition, and also the attachment of the extracellular matrix. So the ECM is the extracellular matrix. Now there are two types of proteins. There are the integral proteins, which extend all the way through the bilayer, and then there's peripheral or extrinsic proteins, which appear on the surface, but they do not go through the cell surface, through the bilayer. So this is a quick picture of what everything would look like. So we see a protein working in transport. We see enzymatic activity happening, signal transduction, which we will focus more on in our cell communication unit. We have intracellular joining, cell-to-cell recognition, which we will also focus in cell signaling unit. This is a very important aspect. And then the attachment to the cytoskeleton and extracellular matrix. So the signal transduction, just to go into depth, it is the process by which a cell receives a signal from outside its membrane and it converts it into a cellular response. So the concept of cellular communication. Now let's look at the different types of transport. We focused on transport, but we didn't focus on the types of transport. So we have passive transport, which does not need energy. And this is when the substance diffuses through the membranes from a high concentration to a low concentration. And nonpolar things, as we talked about earlier, nonpolar things go into the nonpolar lipids easily, and polar and charged things have trouble going through the nonpolar lipids. So small nonpolar molecules such as oxygen and carbon dioxide diffuse easily. Molecules in which cannot be transported easily, we would use transport proteins and facilitated diffusion. So there are many kinds of molecules that do not diffuse freely across the membranes, and the transport proteins provide big enough space for large molecules to pass through. And this doesn't require energy either. Transport proteins and facilitated diffusion, we need to remember, do not require energy. Please keep that in mind. It is something that many people get confused on, but is important. So how does water, which is polar, get through the nonpolar hydrophobic membrane interior? Well, water diffuses through a process called osmosis and moves by facilitated diffusion. And the proteins that are moving water are aquaporins. So, for example, if this was a water molecule, all of these little guys were water molecules, and we knew that, we would also know that the purple transport protein is an aquaporin. Now let's focus on osmosis, which is a diffusion of water across a membrane. Water moves across a selectively permeable membrane from higher water potential to lower water potential. And this process is osmosis. The energy of free-moving water molecules is the water potential. And water potential is the sum of your solute potential and pressure potential. So the formula for water potential is, as shown, And the addition of solutes reduces water potential to a negative value. The application of physical pressure increases water potential to a positive. And then the increased physical pressure can reverse the effects of solutes. A negative pressure reduces water potential on the left side. So here you see a few U-tubes, and these tubes show you an example of solute potential, pressure, physical pressure being applied, and all of the circumstances. So in image A, right here, you see that the addition of the solutes is reducing your water potential. So right here, your water potential is becoming reduced when you add the solute potential. In the image B, you see that the application of physical pressure here is increasing your water potential. So we have the solute potential, which is negative, but then we add the pressure potential, and now you have your water potential to be 0. In number C, or in letter C, we see that we have the pressure potential 0.3, solute potential is negative 0.27, add them together, you get 0.07. And then in D, you get negative tension added. So you have on this side a solute potential 0 and a pressure potential of negative 0.3. Your water potential is negative 0.3. On this side, you just have your solute potential of negative 0.23. Now let's look at how water potential works. We will be calculating the water potential further in the presentation, and you will also need to know how to calculate water potential for the AP exam. Water potential measures the tendency of water to leave one place in favor of another. The Greek letter psi or psi is used to represent water potential. It can be positive, negative, or 0. And the formula is psi is equal to psi p plus psi s. So psi p is equal to the pressure potential, which is a physical component, and the pressure potential of a flaccid cell, which is a cell with less of a pressure, is 0, and the pressure potential of a living cell, which does have pressure, is usually positive, and a dead cell usually has a negative pressure potential. So the solute potential affects the solutes and the effects of the solutes actually. The solute potential of pure water is equal to 0 since there is no solute in that water. This is often referred to as distilled water. Solute potential, if solutes are present, is always negative. And psi s is equal to negative I CRT. The I is the number of particles or ions that the molecule will make in water. For example, so salts would have 2, whereas sucrose or glucose would have 1. So usually in the question they will specify, oh, this is salt, oh, this is glucose, or whatever molecule it is if you need to calculate solute potential. C is the molar concentration from your experimental data or problem. R is the pressure constant, which is usually equal to 0.0831 liters per mole. And then your temperature in degrees of Kelvin. Degrees Kelvin is equal to 273 plus the degrees Celsius of the solution. Water will always move from an area of higher water potential to an area of lower water potential. And if you increase solute concentration, you decrease water potential. If you increase pressure potential, you increase water potential. So just to keep in mind, the formulas that you have for your solute potential, water potential, they're given to you on a formula sheet during the AP exam. So you do not need to memorize formulas. Now I will do an example problem. We'll do number 1 and number 2, since they're both calculating and finding different things. So for problem number 1, we have the solute potential, which is psi s, and psi s is equal to negative 7.7 bars, whereas the pressure potential is equal to 4.33 bars. So what you would do in this problem is you would just do, for water potential, you would do negative 7.7 plus 4. Oopsie daisy. So you do, is equal to this. So I'm just going to explain it. I think it's going to be faster. You're just going to use the formula on the prior slide and add these two up, and that would be your water potential for number 1. For number 2, to find the solute potential of the cell, how we would do this is you know that, your water potential is negative 2.4, and your pressure potential is 4.6. So this would be solved like algebra. And then you would just add psi s. Now plug that into your calculator. You will subtract negative 2.4 minus 4.6, and you will get that the solute potential is 2.2 bars. So it's very logical, the water potential problems, but if you use the information given in the problem, they're not as hard. We have more practice problems. On this slide, you can pause and go through them. I think that would be great practice. We have a few problems on the next few slides as well. And the solutions to all of these problems are going to be on the last few slides. So I can pause, you can pause the recording, I'll stop there, and you can just check your answers. This is another 2 problems. These are a bit more challenging. Now let's look at water balance in these cells and osmoregulation between the cells. So osmosis causes cells to shrink in hypertonic solutions and swell in hypotonic solutions. In isotonic solutions, animal cells are normal, but plant cells are flaccid or limp. They need a bit more pressure. So cytolysis is the cell bursting. Plasmolysis is the cell shrinking or the loss of fluid. And turgor pressure is the water pressure in a plant cell, which is about 2 times the pressure in an inflated car tire. So everything before the slide was considering passive transport. So water potential, osmosis, everything is passive transport. Now we're going to be talking about active transport. Active transport does require an input of energy. Some transport proteins move or pump solutes against the concentration gradient using energy. This is low to high. An example of this is the sodium-potassium pump, which helps communication to the brain. So it's like communication between neurons and brain cells, and the acid-producing cells of the stomach are also a good example. Now let's look at the types of active transport. The sodium-potassium pump, as I mentioned on the prior slide, is a key one. This is when 3 sodium molecules are taken out for every 2 potassiums taken in. And these both go against the gradients. But this is very important to nerve and muscle function. And about 30% of all our energy. Now, in exocytosis, the secretion of macromolecules by the fusion of vesicles with the plasma membrane takes place. And in endocytosis, the import of macromolecules by forming new vesicles with the plasma membrane takes place. So, essentially, what happens is exocytosis uses active transport to take out vesicles. So these vesicles could be filled of, for example, toxins. But endocytosis is to take something in. So some types of endocytosis are phagocytosis, pinocytosis, and endocytosis. So we will explore the different types of endocytosis further on next slides. Now let's look at the exocytosis and endocytosis transporting larger molecules. A vesicle may fuse with the membrane and expel its contents, which is exocytosis, and a membrane can also fold inward and close in the material from the outside, which is endocytosis, and they both require energy. On the first image, you see exocytosis. The molecules are being sent out, but in the second image, the molecules are being taken in, which is endocytosis. So now let's explore the 3 types of endocytosis. Just to recap what endocytosis is, it is the import of macromolecules by forming new vesicles with the plasma membrane. Phagocytosis is when the whole cell engulfs another. So, for example, this is what white blood cells do. And they engulf bacteria or viruses to destroy them. Some strains of the strep throat or streptococcus bacteria have capsules which prevent their engulfment by white blood cells. Pinocytosis is when a cell drinks droplets of fluid. So I always like to think of these two like this. Phagocytosis, phago sounds big, and it is a cell engulfing another whole cell. Pinocytosis sounds small and little, and it is drinking the droplets of the fluid little by little from the other cell. And then we also have endocytosis, which is when specific receptors receive and take in certain materials. Faulty membranes can overload the blood with cholesterol, and harmful levels of cholesterol can accumulate in the blood if membranes lack cholesterol receptors. So there is a genetic condition where high cholesterol runs in families. This is called familial hypercholesterolemia. This is when the receptors of the cholesterol receptors of a cell membrane are not functioning correctly, and it results in the cell not being effectively able to take out the cholesterol from the blood. And so this condition does result in high cholesterol levels. So where does energy come from? The answer is that chloroplasts and mitochondria make energy available for cellular work, and enzymes are vital to reactions which make energy. Chloroplasts carry out photosynthesis, and they use solar energy to produce glucose and oxygen from carbon dioxide and water. Mitochondria consume oxygen in cellular respiration and use the energy stored in glucose to make ATP. This is a short introduction of our next unit, which is going to be cellular respiration, and then after that, we'll explore photosynthesis. So if you would like to see some answers to the practice problems, you can pause the video and check your answers to problems 1 to 6 here and problems 7 and 8 here. Thank you for watching Mathness and More. Remember, if you have questions, please email mathnessandmore@gmail.com, and you can also explore our other AP Bio course series videos in our playlists. These videos will be effective in your prep for the AP Biology exam. Thank you for watching.